I would like to welcome all participants to this webinar on toxicology and pathology aspects for the drug development processes in our industry. Pathologica regards this topic as a fundamental field of expertise in the pathway of many products of drug approval, and has signed an agreement with Patho-Logica for collaboration in these safety trials and consultancy. It is my pleasure to introduce to you the speaker, Dr. Tzadok Reuben, the founder and owner of Patho-Logica. Tzadok did his academic study and work in the USA and recently moved to Israel. He is the DVM, PhD, and US board certified veterinary anatomical pathologist, and in general, and has the same degree, also board certified in toxicology, and a fellow of the International Academy for Toxicologic Pathology. For 16 years, he worked in the pharmaceutical research and development product safety assessment at the GD Serial and CO and Hoffman LaRoche. In 1995, he founded Patho-Logica Consultants, where he has been contributing to product safety assessment and regulatory registration. I'm happy to announce that recently Pathologica and Patho-Logica consults formed a collaboration, business alignment. The topic of this lecture is from conceptual idea to marketed product. I hope you will enjoy, and I hope all of you will benefit from his talk, from this webinar. We will have some time at the end of the session for 2 or 3 questions, and I wish Tzadok all the success. I will now share the screen and call Tzadok to start his presentation. Okay, can you see the screen? Can you see it clearly? Somebody? Hello?
Yes, we can see it.
Yes, we can see it.
Okay. It's better like this or full screen? What should I do?
Full screen.
Full screen. No, it's not good, the full screen. I will come back to this, sorry. Okay, so Tzadok, please find your place and you may start.
So you want to adjust the screen?
Yeah. Yes. I think I will correct it to you. Just a minute.
Excellent.
Yeah.
Greetings to you all. I am very pleased to share this presentation with you. As you can see from the topic, we're going to be having a very complex and long process. So in this short time and in this short introduction, the presentation will be actually highlights of the process. It is our intention to have a longer course down the road, maybe for either a full day or so, in which we will be discussing the process in details. The topics that we'll touch upon today are from the definition of the different disciplines that are involved in safety assessment, to the how to get to some ideas, the history of safety assessment, the different steps themselves, and at the end, all the way to marketing after the approval. And then we will touch upon an example that there could be toxicology problems or safety problem while the drug is on the market or the compound in the market. The process is ups and down, as you can see, one step after another. It is important to follow it one step after another. And what we're involved with actually is in the sciences, the regulatory process itself, the interdisciplinary management between the different aspects that are, or the different fields that are dealing with the product development, and then business, financial, market condition, etc. And we will concentrate today, and in this presentation, actually on this area here on the top. The fundamental disciplines are those of the study of health. And what we mean by study of health is that we're dealing with products that affects the living organism in the context of aliveness and well-being. That's what we mean here by health and the study of health. Toxicology, the science of poison, that's very clear that we're studying the poisons themselves, but in safety assessment, we're also concerned with what the organism is doing to the poison and what the poison is doing to the organism. That's toxicokinetics and toxicodynamics. The same with pharmacology, what the drugs and the therapeutic agents are doing and what the body is doing to them. Pharmacokinetics, pharmacodynamics. And in pathology, we're dealing with the study of disease, functional impairment, and very important to realize that in the study of disease, we also study recovery from the disease, not only the pathogenesis itself and the causes of the disease. There is in it the morphology, the clinical laboratory pathology, and a lot of research, which we call, for this case, for our case, investigative pathology. It is important to realize that toxicology by itself is a bit different, or greatly different actually, from product safety assessment. An expert toxicologist may not have any familiarity with product safety assessment that we talked about, the process itself, the regulatory registration, and the going into the market. What we are interested in, the basic thing in product safety assessment, is we are determining if the product is safe, that it is accepted for regulatory approval and marketing in the context of the circumstances that it is used. For example, thalidomide, a very terrible toxin, now is used with specific precautions for leprosy, cancer, and is being investigated for other drugs, for other diseases, I'm sorry. So product safety assessment has its history, and that led to the good laboratory procedures, to the GLPs, and with that to the good manufacturing procedures, the GMPs, and the good clinical procedures with the GCPs. Prior to thalidomide in the 1950s, 60s, not very much attention was paid to the side effect and to the adverse effect of therapeutic agents. Classically, mercury was used as a therapeutic agent. Now, thalidomide, when it came in and what was found with it, that was a breaking point. From there was a lot of attention, especially in the United States, with regard to the safety of the products that are used for health. And it entered into the law of the land. Congress in the United States put the rules for the good laboratory procedures. Now, prior to that, and what pushed into that was that in the about mid-70s, there was an inspection, an FDA inspection of one CRO, and what was found there, what the inspector found, that the date for withdrawal of blood for hematology was later then the date recorded for the death of the rat. And this is what's going on here. Things got very involved in the inspection of that laboratory. They found a lot of faults in it. The laboratory was closed. Its owners and senior people there were penalized legally. And from there, it was even more enforced with the whole this area of the GLPs. And in it, the emphasis is on quality assurance and the designation in the law that the study director is the person responsible for everything in the study. A toxicologist, most of the study directors are toxicologists, but it's not necessary that the person will be a toxicologist or a pathologist. It has to be a person who is responsible. Then developed whole area of the peer review and the necessity for peer review, because pathology is a semi-quantitative science, and we need to have an opinion of an additional pathologist. And after all that, and just in the last 15 or 20 or 25 years, the GLPs are now international. Now we come to the product itself. So we need to have sources. Where does the conceptual idea is coming from? And you can read, and these are only just very few examples from which an idea could come to develop a product. I will take just a few of them. Observation in nature, cafe and caffeine. Many, many years ago, 100 years ago, shepherds of goats in the Sudan and Ethiopia saw when the goats were grazing in areas in which the cafe shrubs and trees, when they're bearing the beans, when they were grazing on them, they got excited and moved very, very more like almost erratically moving. So they said, aha, maybe we could have a stimulant here, and that is the cafe and the caffeine now. It could be incidental in the laboratory, and the classical example of that is the penicillin that was kind of thought about by an error, supposedly. And another example is aspartame, the sweetener, so just about 40 years ago or so. It is a natural dipeptide, and the chemist who was working with this peptide, being it natural, he says, what's the big deal? I'm going to taste it. And when he tasted, he found out that it's sweet. From that, developed a sweetener. Of course, we synthesize. Biological lead is the main thing. A classical example of that, it was found out that histamine has to do with allergies. So then let us find an antihistamine or the antihistamines. We can induce, and it's possible to induce chemicals in the living organism. For example, we give tetanus to horses. The horses develop immunity. We take their serum and we have an anti-tetanus product. Then, of course, the issue of all the implants, that they are either from natural parts or they're artificially made. And we can use, and being used, organisms for delivery as drug carriers. Viruses, for example, you could put on them a chemotherapeutic agent for cancer. You are gaining targeted delivery, less drug, less toxicity, more efficacy. Now we come to the discovery process or the research stage, which is leading to development. The key issue here and what our goal is to find a clinical candidate from many, many candidates that they are saying we have about 15 analogs of the same chemical. Which one is the best? So we have a design. Design is different than a chemical synthesis. We synthesize. We put it through a process or processes of primary and secondary assays, a lot of in vitro, sometimes also in animals. We sometimes do structure-activity relationship between the different analogs to find out which one is the most effective, its relationship to a ligand protein, and from there, we have a clinical candidate. Wow. Research now reached a very, very important stage. We can send it into development. And within development, The most important thing is the toxicological evaluation. For the toxicity study, we would want to have a protocol and be concerned about amendments and deviations from the protocol. You can read here, and some of you probably are familiar of what it will take, the title, whether it's a GLP or not. The important thing about it is to have a clearly defined purpose of the study, its details, materials and methods, and where it is conducted, and references. These are all. Now, the study director's signature is a must, and some companies require additional signatures, say for the supervisor or the vice president or the director who is responsible for the entire area. A very important point is the extent of the details that are being mentioned here. We do not want to have too many very intricate details. We need to have a gray area, use judgment in order to avoid amendments. On the other hand, we don't want to be too loose. The most important thing is not to have deviations. So the SOPs to be followed and the rules of QA are to be followed, a must. What are included in general toxicity studies? The in-life, which is the clinical stage, in this case of the animals, as you know, general observations, food consumptions, etc., or ophthalmic, and these are the main, and others as needed. Clinical pathology, pathology itself, necropsy, and histopathology. That is usually included. Study report. Okay, again, the title, the signature place, the study number, etc. The most important thing in it is the summary. The summary, it should be concise and clear, and it should be easy to read and to understand and to get by the reviewer or other people in the company who are not the scientists. We do not want to get into a situation where a reviewer, especially starts to question and wants to go to every detail in the tables inside the report. Sometimes they do that, but we want, if the summary is clear, there is no need for it. Then there are the findings themselves, the pathology report, other related report, and to the study report. Animals that are being used, the rodent, which is mostly the rat, but also the mouse in carcinogenesis, and the non-rodent is the dog. Another non-rodent is the non-human, the cynomolgus monkey, because it is smaller, easier to handle in the laboratory, and it's we need to use less. We can use a lesser amount of drug, especially at the beginning when synthesis is very limited, and its genetic makeup is a lot more defined than cynomolgus monkeys in the wild. There is the rabbit in teratology, the guinea pig in skin sensitization. These are the main things that we encounter in them. Then there are the others that you are reading here, the others including modified humanized mice, and I put cell culture because still cell culture is being in toxicology quite a bit. The types of general toxicity studies and the standard are the single dose, the repeated dose up to 1 year, with interim programs, interim groups, reversibility groups, oral and intravenous again. But then we have the standard genotoxicity with its 3 tests, reproductive with its types, and skin hypersensitivity and the carcinogenicity study. The types are those that are a little bit special or unusual. Now, I would like to tell you something about the hair. The hair is an in vitro test that to test for the action potential of the heart muscle cells, and we are concerned with potassium channel, especially with prolongation of the QT interval. It has been found that this is a major thing that causing with arrhythmias. That is an fairly easy test. It is short time and it is relatively inexpensive. Very, very useful information. And then there are the others, including pediatric toxicity that can be done. Now we come to the preclinical phase itself. And we need to differentiate between preclinical, which is before the clinical in animals are being used, and along the clinical trials in humans, we continue to do non-clinical studies in animals. We want to reach the IND, the investigational new drug, and its regulatory approval, which allows the first entry into man. This is a milestone stage. It is a very important stage within the company itself, because it is moving now to clinical trials and further development with a lot more investment, money and time. But it is also very important for startup companies, because then they have a product that they could show for which they could get either financial support or being acquired by a larger company. Now we go to the studies themselves, and we start with a single dose for which we would want to select the doses for the repeated doses that will follow. We're interested in avoiding lethality in the repeated dose studies. We don't want animals that are dead. We want to examine the animals at the end of the study when they are still alive. Okay, we do oral, we do IV, as you know, in the rat and the dog. In the rat, we do all the doses at the same time. In the dog, we have very few animals, so we do escalated dosing. And we usually do not do necropsy, but in the dog, we might do, especially if they are dead. The preclinical phase now starts. 2 week. Some companies do non-GLP study 2 week. The advantage of that is that there are limited tissues that are being taken that could tell us a lot. Less time, less money. Why is it 2 weeks? 2 weeks legally with the IND. If you have a drug or a compound which is used only once, one dose in humans, a 2 week study GLP toxicity can clear it from standpoint of regulatory approval. I had an experience with that. It was a colonic cleanser for colonoscopy. In animal study, general toxicity, the 2 week were sufficient for the one dosing in humans. Then again, we do the oral and IV, rat and dog, clinical, etc., etc. And we mentioned that the histopathology is limited. Some companies do early genotoxicity studies, especially the Ames test, and some of them also do the hair. These are because they are in vitro, very quick, give a lot of information, and they are relatively inexpensive. Then we come to the GLP itself for 2 weeks, and then we could go usually in the interim for 13 weeks. Now, why 13 weeks? Because 13 weeks is going to give us a lot of information for dose selection, for the longer up to 1 year, and for the carcinogenicity study that can be up to 2 years or 1.5 years. So that is why the 13 week. Also, the 13 week allows us to go to more than one dosing into humans. Here, the histopathology is full, and there is a peer review usually. The peer review is not required by law. It is very good to do, and we don't want to avoid it. Now, since we have now all the information behind us, we want to go to the IND submission itself and its approval. It is including the pharmacology with all the information about it, with all its summary, including literature, etc., which we talked about before as far as the research. The toxicology summary, the most important thing in it, again, is the summary. The reviewer goes right to the summary and wants to see what was found and the definition, what is the NOEL and the NaOL, the no effective dose, and the level in which there are no adverse effect levels. Very, very important. Sometimes there is also some information from ADME, which is the absorption, distribution, metabolism, and elimination in animals. And in the clinical now, the doctors are preparing now their clinical part for the IND, in which they are putting a proposal for escalating doses in human recipients, and if that, and I'll show later, if that's accepted, goes further to people that are carrying the disease. There is a meeting with the FDA. Hopefully there is an approval. Bad news if there is no approval. And here's an example of the compound that I was involved with. I will come to it later. It's an cardiac antiarrhythmic. It hit the clinic, but it passed the IND approval. Now we come to post the IND, the NDA, the new drug application. Now we do non-clinical phase in animals, etc., along the clinical trials, which are phase 1, 2, and 3. You heard a lot about it now recently in the past year, phase 1, phase 2, phase 3, with the vaccine that is being developed for the COVID-19 virus and the infection. In phase 1, it is in healthy subject, limited number. We do escalated doses. So for example, the proposed dose will be 10 milligrams. In human, we start with 1. If everything is okay, we go to 3, we go to 6, then we go to 10. And if 10 is still okay in humans, we go a little bit higher in order to decide and to determine what is the highest tolerated dose that can be used later and very important in clinical use. In phase 2, if everything is okay, we get approval for phase 2. Phase 2 is also limited number, but these are subjects that carry the disease. So here we are also looking for the response and observation as far as therapeutic is concerned. Some ADME is being done, or more ADME is being done in phase 2, leading us to phase 3. Phase 3 is the real critical, large number, as much as possible, as reasonably possible, as scientifically reasonable, in order to determine if the drug is working, efficacy, is tolerated, toxicity, and if it is manageable and easy to manage. It is big submission, a lot of trials, and from there, we're hoping to have we're also going to be the non-clinical, in which we are going to do the general toxicity studies up to a year, sometimes with an interim. Again, dog, rat mainly, histopathology, full tissue. Now peer review is mandatory. Further, non-clinical, the carcinogenicity study, which is the largest and the longest, takes time and the costly. We want to start it as early as possible because we don't want to get that clinical phase 3 finished and we are still not finished with the carcinogenicity study, with its histopathology, the peer review, and the report writing. We don't want to get into that, but we don't want to start too early either. There are some potential supportive alternative studies or systems for carcinogenicity studies. They are not acceptable as far as the FDA, but they are being worked on, especially humanized mice. And then the non-clinical should complete all the genotoxicity, all the reproductive, all other systems, all the special toxicities that are needed, and we are ready for the IND itself. The IND itself, the submission, includes the NDA submission. It includes the IND that we talked about before, and here there is again summary. The pharmacology, the toxicology, the ADME in animal, the non-clinical, the clinical trials with their ADME, a discussion, references. Hopefully, we are going to have a meeting with the FDA, and hopefully, more hopefully, that there is an approval. Here is an approval with a compound that I was involved in. Big celebration for the company. There could be a problem post-marketing. The drug is on the market and we find issues in toxicology. Spironolactone was antihypertensive. Someone, a laboratory in Japan, did on it a carcinogenicity studies. They found that it induces tumors in rat. Searle, that was marketing the spironolactone, was notified. They asked the FDA to give them time because there was no indication prior to that of carcinogenicity in humans or in animals. They did a metabolic research. They found out that metabolism in the rat is different than that of human, and the metabolite in rat is not associated with carcinogenesis. The matter was cleared through today. Spironolactone is on the market. Then there is this issue, which is that liver cells in the rat, with in toxicity studies, the inner cells in the lobule gets larger than those that they are in the periphery. This is a very, very common and well-known phenomenon. It is adaptive because the proteins in these cells are much more and the cells are larger, but it was found out and suspected that it's associated with carcinogenesis. Hoffman-LaRoche, in collaboration with major toxicology institutes for research, did major studies on that for many years with a lot of money, and it was found out that this phenomenon does not have to do anything with the carcinogenesis in the rat. That is one type of investigative pharmacologic toxicologic pathology. Another case is disobutamide. This is the antiarrhythmic compound that I mentioned before. It causes these vacuoles inside the cells in the coronary artery of the dog. The drug was in advanced clinical stages. It was dropped, and it was decided to do research. What is the nature of these vacuoles, and can we avoid them? It was work of the pathology, toxicology, pharmacology, and the chemist was very important. We found out that the key issue is this area of the amine of the nitrogen on the compound that got changed, but this area of the molecule is still having an antiarrhythmic activity, and that was the finding of the new drug that I mentioned before, that antiarrhythmic drug that you saw on the cup that hit the clinic. So in summary, the process, as you know it, is very complicated, is long, and it is exciting especially when we have good results. And I could tell you from experience of 16 years being employed in the industry and more than 25 years as a consultant, is that it is a wonderful area, wonderful specialty, and very exciting, a lot of contribution to both to health and mankind. And I wish you that you will have the same excitement that I have had. And I thank you for your attention. And here we are. Thank you.
Maybe somebody would like to question.
If you have questions, I'm open to questions. I hope.
Well, if there's no question, maybe I can ask you a question, Tzadok.
Yeah.
Maybe you can give some information regarding cell therapy. This is a quite hot topic these days, and I know that the authorities are very concerned about stem cells and cell therapy. What do you know? What do we know about these kind of therapies regarding proliferation of tissues and the risk for neoplasia specifically?
First of all, it's a different area of so-called standard toxicology. I'm talking about standard toxicology is what we saw before with the animals and things like that. A lot of biochemistry, a lot of cell biology, a lot of in vitro, a lot of genetics and genetic biology. But the main concern about it, what is being done with this cell that is taken from and it's being modified, some modification into it, and being put back into the recipient. Whether these changes, especially the genetic changes that could occur in it, are now going to be adverse effect in the recipient. And the question now, how to go about it in a non-clinical fashion? That's a tough issue. It's a tough problem. Then probably the best to do is to take a genetically totally inbred mouse, to take its cells that is comparable to that of humans, do the modification in it, back to the mouse in order to see if there are any bad activities or not. And still they'll tell you, how do you know? A lot more. We don't have sufficient information about it. However, it is a very important field, and now it is becoming a major field in both therapeutic ways or therapeutic methods, but primarily for safety assessment. It is involving a lot more interdisciplinary. And I must tell you that the scientists who are great scientists in this area, okay, are very, very frustrated by what is required for safety assessment, regulatory requirement, and regulatory approval. They are very frustrated. They are so involved, and their minds, the great minds, are involved in it, and they just are frustrated. They conceive why it's very clear to us. It's not very clear to the regulation, to the regulatory process. And it is very dangerous if safety is not being taken into account. Any other questions that you might come up with?
No, then thank you very much.
Thank you very, very much for your attention. Let's be in touch. Thank you.
Okay.
Thank you.
Thank you very much, everyone. So you saw the details of Patho-Logica and Patho-Logica.
Toxicology medicine.
Thank you.
Thank you very much.
If you
Manu,
would like to follow up, no problems. Yes?
can you hear me?
Yes,
will
yes,
it be
Ronnie.
possible?
Yes.
Will it be possible to get a copy of the video for colleagues who could not attend the lecture?
Yeah, I think it was recorded, and we can provide this. No problem. Yeah.
Thank you very much.
You're welcome. Yeah.
We look
Thank
forward
you.
we look forward for you in the more extended course, because then we could take a lot of cases and we will take them more like a case report and analysis, what was going on, what we went through, how it got resolved, and how it did not get resolved until today.
Okay, so thank you again, everyone, for participating in this exciting webinar, to my opinion. And feel free to contact me or Tzadok directly if needed. Thank you.
Thank you very much.
Yeah, bye-bye.
Thank you very much. Bye-bye.